everyone, for bearing with us. Sorry for the delay. Good news that we had some buffer time there. I'll probably still get done by 4 as originally planned. So I'm Ben. I'm going to be talking about personalized regression and how we can use this for sample-specific pan-cancer analysis. This is now in PDF format, so please don't get scared when equations come up. I intended to show them one at a time, but they might come up as a whole slide. That's okay. So we all know that cancer is a very complex disease, and that's why we have to do all these data analyses for it. In particular, we have different mutations causing similar or different phenotypes, and there's many, many different possible driver mutations. So as we go about building all these models, we're tempted to build bigger and bigger models that explain more of the variation. But maybe we don't really need to build one single model that explains all cancers, because all cancers are very diverse. What if we could instead build a different model for each type of cancer? The downside to this is that cancer type is unknown. It's not annotated as any clinical covariate, even as some tissue may be. So if we took this idea to the extreme, maybe we could estimate a single model for every sample in our data set. This would correspond to trying to understand tumors one at a time. And this would allow us to use simple models that each work for a single patient. And then once we have these single patient-specific models, we could ask new types of questions like, how does one patient's tumor differ from the cohorts? So graphically, our goal is going to look something like this. And that is a sample-specific pan-cancer model matrix. Each row in this matrix corresponds to the model for one patient. The entries are model parameters. So you see that each sample has a different model parameter, and there's not necessarily any mixture model that defines the contribution to the different rows. And once again, why do we care about building sample-specific models? Well, in machine learning, there's this trade-off between effect complexity versus effect personalization. This is all assuming that we're looking at some complicated phenomenon. If we believe on this left side that there are universal but complex effects, then we should probably use some complex model that is estimated for the entire population. So for instance, if we're doing a self-driving car project, we believe that driving is a pretty complicated task, but every self-driving car should act in approximately the same way. On the other extreme, we have cancer. And we believe that cancers are relatively simple, but they're highly personalized. So maybe there's only a couple driver mutations for each part of a tumor. Of course, we know there are clonal populations as well. But we can imagine that it's much simpler to think of sample-specific explanations than to build a population model. In the middle of this gradient is mixture models where we assign each point to a cluster, and we can estimate some possibly complex or possibly simple model for that cluster. And why do we want to do this in a pan-cancer way? Well, here I'm showing a table of the number of samples in TCGA. And we probably all know the Cancer Genome Atlas, but for those who don't, it's one of the largest public data sets for cancer genomics. And we see that for the common ones, there's more than 1,000 samples. But for the rare types of cancer, there's fewer than 50 samples. So it can be really, really difficult to learn meaningful models from these small sample sizes. But if we can learn pan-cancer models, maybe there are mutational signatures that span the tissue types, and so we can share information between the common and the rare types of cancer. So there's a lot of related work trying to estimate specific models. The variant coefficient model of Hastie and Tibshirani is nice, but it assumes that there is some parametric form which translates clinical covariates into model parameters. There's also been some nice work on known structure that's been presented at ISMB in years past, especially on tree-structured or time-varying models. And there's also been more recent work on sample-specific network estimation. But these are generally formulated as, for network estimation. They're not really general frameworks. So here we're trying to present a general framework for estimating sample-specific models. We're going to present this in the context of a linear regression, although as you'll see later, this can generalize to more complex predictive models. So on the top, we're showing the standard form of a linear regression. This would be a population linear regression. So we say that the y vector is x times beta transpose. Beta here is the population effects. We're assuming that there are the same effects for everyone in the population. In contrast, we're going to estimate yi in terms of xi and beta I. So beta I is our sample-specific model, the parameters for this one person's model. And again, graphically, that's going to look like over here. So we have this row is going to be beta 1, this row is beta 2, all the way down to beta n, where n is the number of samples that we observe. Of course, this is a highly overparameterized problem. So machine learning people are probably going to look at this, or statistics people, and say, well, it sounds nice, but you're estimating a single point here for an entire model. That seems kind of crazy. We can induce, as it turns out, we can induce structure in the model parameters according to the model covariates, and we can actually recover this pretty well. So here, I apologize, it's a little overwhelming slide, but to do this, we're going to define a sample-specific loss function. So this is our standard loss function, L. And we are instead going to use this overall loss function as the sum over n different sample-specific loss functions. These sample-specific losses are composed of the standard prediction loss, a standard regularization loss, and a new distance matching regularization. So if we were to just use these first two items, which would be how we might go about estimating sample-specific parameters, it would be incredibly overparameterized and we'd have no hope. But since we induce a new distance matching regularization that promotes distance in model parameters to be similar to distance in model in sample covariates, then we can actually induce structure in our model matrix and recover meaningful parameters. So distance matching regularization follows the idea that the distance between sample parameters should be similar to the distance between sample covariates. So for sample I, the distance matching regularization is a function of d beta and du. D beta and du are distance metrics. And it is the sum over all pairwise distances, so the neighbors j, and the squared error between the implied distance in parameters and the implied distance in covariates. So when we optimize this, what we're going to end up doing is pulling together parameter distances and covariate distances. Now, how do we define these distance metrics? That's a pretty tough challenge. And in fact, it's, if we could do this a priori, maybe we should go with a varying coefficient model instead. So we instead want to let the data define our distance metrics. We're going to use linear combinations of feature-wise distance metrics. So for d beta, that is our distance over model parameters. We're just using the L1 norm, but it's weighted by phi beta. And for du, the distance over covariates, we have user-supplied feature-specific distance metrics. So here we're assuming that we have k different clinical covariates, and the user has given us a feature-specific distance metric for each one. Then we're going to optimize phi beta and phi u, and we can inspect the values in those vectors to understand what our personalized models are responding to. And of course, the user has to supply these individual distance metrics, but that's actually quite nice. So you can imagine arbitrarily complex distance metrics. For instance, if you have a covariate that's an image, maybe you have some deep learning-based distance metric. So once again, personalized regression is a model for inference, retrospective analysis, not necessarily the most accurate predictive model. We also believe that they're relatively simple personalized effects, not complex universal effects. If we're trying to understand complex universal effects that have been, for which our observation of them is perturbed by covariates, maybe a mixed effects model would be more appropriate. We also need to have covariate data, which is informative of each sample. And if we know enough about the covariate data to write out a predictive model, there are many models for that. Instead, we're assuming that the covariate data is informative, but we don't know exactly how. So for this analysis, we focused on TCGA, and we come back to this table and the number of samples. So we're doing a pan-cancer analysis of TCGA, and our task here is to predict case control status of each one of the samples. It's a little bit of a simple task, but what's interesting is that we can learn meaningful models on this simple task. And since our model has been formulated as a regularization term added onto a sample-specific predictive model, you can imagine many ways that we can extend this to incorporate more complex predictive models. Our data comes from 28 different primary sites, about 10,000 samples. We have 4,000 RNA-Seq features and 14 clinical covariates. The clinical covariates include tissue features, sample molecular biomarkers, patient demographic features. And one of the nice things, again, is that since we are operating directly on pairwise distances, we don't really care what the form of these covariates are. We can use covariates directly that are strings, that are unordered, anything, as long as the user has defined an appropriate distance metric. In contrast, traditional methods that write a predictive model on top of these covariates expect the data to be one-hot vectors, which in this case expands it to about 70 different features. So the first thing we see after training these models is that the personalized models are more efficient with variable selection. Over here, we see that the models select fewer genes per sample. So the blue is a histogram of the number of features selected in each sample, where a sample also corresponds to a single model. And in the red lines are the number of variables selected by a population model trained on one specific tissue type. And we see that the tissue-specific models select almost all of the RNA-seq features, but the sample-specific models get away with only selecting about 500. And in addition, these genes that are selected for the most part, fewer than 500 samples. So this supports our hypothesis that the reasons for cancer may be relatively simple, but they are extremely personalized. So building one single model for the entire population is going to be difficult, if not impossible, but building maybe sample-specific models is possible. We also look at the selection of oncogenes and tumor suppressor genes. So here I call it an oncogene if it was annotated in the Cosmic Gene Census, 2014. And what we do is we rank the variables by the variable selection. So a coefficient is assigned to each variable after optimization. And for the sample-specific models, we sum the coefficients across all of the samples. So this gives us a population scale estimate of the importance of each feature, which can be directly compared to these population models. And in this plot, what we're showing is the number of oncogenes selected at each rank. So we want to see plots up here, which will imply that our models are selecting as important features the known oncogenes. And what we see, so our method is this red line, the personalized method. The dashed gray lines are the population models trained on single tissue types, which have a weighted average of this black line. So we see that down here, a lot of the methods effectively identify common oncogenes. This is encouraging. That means that the common oncogenes maybe have strong effects, maybe have been studied a lot, and all methods identify them well. But as we get up to here, so this is about 1,000 total genes, 400 oncogenes selected, we see that this dark green line is the population model. So if we train a single population model, it stops identifying oncogenes any better than random after about 400 oncogenes. In contrast, our personalized model continues to select oncogenes, and we hypothesize that's because it's able to identify the oncogenes that are really only important for a couple of patients. So personalized regression also gives us this nice picture. We're returning to that graphical picture of the matrix of model parameters. Here we are plotting the variable selection. So a pixel is dark if that variable is selected for that sample. It is white if that variable is not selected for that sample. And it's colored based on the primary tissue type. Again, we have these red lines down here, which indicate whether that column is annotated as an oncogene in the cosmic census. So we see, firstly, that there is dense selection of variables where there are dense amounts of oncogenes, and sparse selection of variables where there are not that many oncogenes. But the patterns overall don't really jump out to us immediately. So instead we go ahead and cluster. This is a little tough to tell because it's covered up, but we cluster this matrix, and we immediately find some patterns that jump out of this. If you're interested in this figure, it's a lot easier to see in the paper, or I can show you afterwards. But one of the interesting things is there's this small patch of variables that are selected consistently for about 300 patients, but not selected, or selected very, very sparsely for the rest of the patients. And this small set of genes ends up being significantly over-enriched for a particular Go term, which has to do with ion transport processes. And ion transport processes have been seen important in a couple of particular cancers, but maybe not seen as a pan-cancer marker. And here we're seeing that the patients for which this is selected, they span all the different tissues. Finally, we cluster the models. So this is a t-SNE plot of the different of the models. And firstly, we see that it forms some nice clusters. But the clusters, once again, do not correlate with any of the primary tissue sites. So this implies that we're looking more at molecular signatures than at the tissues. And we run, again, enrichment tests for each one of these individual clusters. So we're wondering here, are these clusters actually meaningful? Are we looking at different processes in each one of these clusters? And the answer is yes. So these are the top 3 enriched leaf terms in the Go analysis. And we see that not only are they selected for oncogenic processes, the oncogenic processes appear to be pretty different in different clusters. So in particular, cluster 3 is over here, has a lot of terms that have to do with antigen presenting processes. Cluster 5 has to do with membrane processes, and cluster 8 has to do with the cellular metabolism. Finally, we can analyze the distance metrics that have been learned to get some sense of what is contributing to this personalized behavior, or what we have learned may be contributing to the personalized behavior. So here we're seeing a contribution to du. Du is our distance metric over the covariates in terms of each one of the covariates. So we see the most contributing site, as we may expect, is the primary site and the disease type. And then we see this variable days to collection, which probably has to do with batch effects in TCGA data. Then there's some demographic features of the patient, and finally molecular markers of the tumor itself. So while this picture is nice to look at, one note of caution, the difference between the most important and the least important clinical covariate is only about 30%, right? And this is actually, this supports our idea that all of the covariates are actually meaningful. We can't really split this data along any one covariate and come up with a meaningful model. Instead, we need to take them all at the same time. So in conclusion, I presented a way to estimate sample-specific models. We call this personalized regression with distance matching regularization. And when we apply it with this simple idea of logistic regression for pan-cancer transcriptomic analysis, we see a bunch of clusters that are not found by traditional methods. We're continuing to work on this. So this has to do, we have a lot of biological questions coming from that matrix of model parameters. So are these sample-specific processes that we saw, are they real? Is there some molecular marker that could be meaningful clinically? That still needs to be investigated. Of course, we also used a very simple task, a very simple model, case control identification with logistic regression. But since we presented this as a regularization technique, we can actually incorporate any personalized model. And finally, I'm also working on applying this to single cell data, election modeling, and stock prediction. If you're interested in any of those kind of obscure, well, not obscure, but different topics, please feel free to talk to me after the break. So in conclusion, thank you everyone for listening. Thank you for the organizers of ISMB and CAMDA. Thanks to my collaborators, Brian and Eric. We have code here on my GitHub. So as I said, this is presented as a regularization technique for any model. This means that our Python code here takes in a predictive model as a function and then can optimize that black box function to produce sample-specific models. Thank you again to ISCB for the travel support and for the NIH for supporting the research. Take any questions.
Thank you very much, Ben. Do we have some questions? Yes, please come to the microphone.
Great talk. I would like to ask, what do you know about metastasis? A cancer moves to another location. How do you model it?
Yeah, so the question is about how do we model cancers that have metastasized?
Yeah.
Yeah, that's tough. So I guess we're continuing to appreciate the complexity of cancer. It's not just like one disease in a patient, but then there's a mosaic, right? I don't know that the data is there in TCGA to be able to model that, but if I had a data set which somehow could, I'd be very interested in that.
Yeah, please just come to the microphone.
Backtracking what you have done, you have concluded that all the covariates had meaningful contribution towards it, the model. If that is the situation, why do you even bother for regularization? Why can't you do the Latin variable modeling like principal component regression or partial least square regression?
So if we need all of the variables, then why do we want
Regularization, I mean, why do you want to?
So I mean, yeah, that's an interesting question. It is hard to interpret when we say that all of the covariates are meaningful, right? I think it's very difficult to write a model which takes in all of the covariates and it gives another model, which is, in a sense, what we've done here. But instead of actually writing out the parametric form of that, we allow it to be optimized away from the personalized or from the population model.
Yeah, but any of this principal component regression or partial least square regression, they will do the job for you. They will fit the optimal model. I mean, so something to think about.
Sure, yeah. Let's talk.
Very interesting approach, and I'm just trying to model, to map your model on my mental space, and I'm just thinking if it is just that you have a regression towards these general effects, and then you add these variables that are more extremely different in particular patient from the norm, I would say. Is it this what this regularization, this second regularization do?
So keep in mind that it also allows us to get rid of the common effects for the population.
Yeah, so this is what the basic part of, and then you have still this part that is more personal, yes? And isn't it just finding these genes that are specially different in particular patients from the norm, yeah? Does it work this way?
So you could think of it that way. Although, if we simply look at the if we just look at the data and do a clustering analysis on the gene expression, we don't find any of the same enrichment patterns. So here I'm showing the similar t-SNE plot that I showed for the models. And what we find is that because we learn the models rather than just looking at the highly enriched genes for each sample, we have more structure in the data, and we actually get clusters that are enriched for Go terms. Does that answer your question? Talk offline.
Okay, thank you.
In single cell, we are suffering from a lot of zeros. And your model, can you handle this at this point?
Zeros in the data or zeros in the covariates?
Zeros in the data.
Yeah.
Because you are a lot of genes, it's not expressed in this cell. And
yeah, so that's an interesting question. And there's two parts, right? So one is if we expect the zero to be a real biological phenomenon and we want to model that, then I would say that yes, this could handle it well. But if we expect the zero to be a technical artifact, maybe it will be difficult to estimate a sample-specific model.
So you motivated this bank cancer genomic approach for cancer types where we don't have data. So for cancer types where we have enough data, did you try to apply your model to just data from this type?
So yeah, I think for the TCGA data, the most common one is the breast cancer, which has about 1,000 samples. And the breast cancer is actually one of these lines here. So even in this one particular tissue type, we're not able to select a small set of genes that are meaningful for it. Now, it's possible that there are more clever approaches that could be used to do that. But here, if we just use a simple logistic regression model, we're going to end up selecting almost all of the genes, even for one.
No, I mean you still build a personalized model, but use the samples only from breast cancer.
Oh, no, I haven't tried that yet. That would be interesting.
That's what I meant.
Yeah, okay.
Okay. Thank you very much once again. And